Welcome to this deep dive into the fascinating world of microbes. You know, it's amazing. You've sent over such a collection of articles and research on everything from like the tiniest viruses to the complex world of fungi. It's a lot to unpack, but I'm ready to kind of dive in and explore this hidden universe.
It really is amazing how much is happening right under our noses at a scale we can't even see. These microbes are everywhere, shaping our lives and the planet in ways we often don't even realize.
Yeah, you're right. You know what? I'm particularly intrigued by the microscopy material that you sent. Before we even like get into the different types of microbes, I'm curious, how did we even discover this microscopic world?
Well, microscopes were, and still are key to unlocking this invisible realm. They're like windows into a whole new dimension. And you've got information here on several different types of microscopes, each with its own unique way of revealing the microcosmos.
Yeah, there's so much variety. You know, we've got the classic light microscopes and then the electron microscopes, which you mentioned can magnify things thousands of times more. And even these newer fluorescence microscopes. It's like we keep inventing new ways to see the unseen.
It's like we're constantly upgrading our vision, going further and further down the rabbit hole. And each type of microscope gives us a different perspective. For example, the electron microscopes, like the TEM and SEM you mentioned, use beams of electrons instead of light, which allows them to see things at a much higher resolution.
So what kind of details can we see with electron microscopes that we'd miss with a regular light microscope? Can we actually like see the structures inside a cell?
Absolutely. The TEM, or transmission electron microscope, lets us see inside cells with incredible detail. Imagine being able to see the intricate structure of a mitochondrion, the powerhouse of the cell, or the delicate folds of the endoplasmic reticulum.
Wow, that's like having X-ray vision for cells. And what about SEM?
Yeah.
What does that show us?
The SIM, or scanning electron microscope, gives us these amazing 3D images of a specimen surface. It's like being able to explore the landscape of a microbe's outer shell. You can see every bump, every crevice in stunning detail.
That's incredible. So, okay, we've got these amazing tools to visualize this microscopic world, but let's talk about the stars of the show, the microbes themselves. I see you've included a lot of information about the differences between prokaryotic and eukaryotic cells.
Yes, that's a fundamental distinction in biology. Prokaryotic cells, like bacteria and archaea, are the simpler, more ancient form of life. They lack a true nucleus, so their DNA is just floating in the cytoplasm. Eukaryotic cells, on the other hand, have a membrane-bound nucleus to house their DNA, plus a whole array of other specialized compartments called organelles.
Organelle. So are those like tiny organs inside the cell, each doing a specific job?
Exactly. Think of a eukaryotic cell like a bustling city. Each organelle has a specific role to play in keeping the city running smoothly. You've got the nucleus, which is like City Hall, containing all the blueprints and instructions. The mitochondria are the power plants, generating energy for the entire city.
Okay, I'm starting to get the picture. What about the lysosomes? I remember those being mentioned in the material. What do they do?
Lysosomes are the recycling centers of the cell. They contain powerful enzymes that break down waste materials and worn-out cell parts, keeping the cell clean and functional. And this process of cellular recycling, called autophagy, is really fascinating.
Autophagy. So is that like the cell's self-cleaning mechanism?
Exactly. It's how cells get rid of damaged components and recycle the building blocks to make new ones. What's really interesting is that research suggests autophagy plays a role in aging and even disease prevention.
Wait, so keeping our cells clean on a microscopic level could help us live longer and healthier lives. That's incredible. Okay, back to the cell city analogy. We've got the nucleus, the power plants, the recycling centers. What other essential services are happening inside a eukaryotic cell?
Well, there's the endoplasmic reticulum, or ER, which is like a network of highways and factories involved in protein production and transport. Then there's the Golgi apparatus, which is like the post office, sorting and packaging proteins for delivery to different parts of the cell, or even outside the cell.
So it's like a highly coordinated system, with each organelle playing a crucial role. But what about plant cells? Are they fundamentally different from animal cells?
Plant cells have all the same basic organelles as animal cells, but they also have some unique features. For instance, they have a rigid cell wall made of cellulose that provides structural support, allowing plants to stand upright and reach for the sun.
Ah, that makes sense. Plants need that extra support to grow tall and withstand the elements. What else makes plant cells unique?
The most striking difference is the presence of chloroplasts, the organelles responsible for photosynthesis. These are the green powerhouses that capture light energy, and convert it into chemical energy in the form of sugars, which the plant uses for growth and fuel.
So plant cells have their own internal solar panels. That's amazing.
Yeah.
Okay, we've covered a lot of ground here talking about the tools we use to see microbes and the basic differences between these tiny
It's
cells.
just a glimpse into the complexity of this microscopic world, and we haven't even touched upon the viruses and prions yet, which challenge our very definition of life.
I'm definitely intrigued. But before we get ahead of ourselves, let's talk about the molecules that carry the instructions for life. DNA and RNA. I see you've included some information about their differences.
Both DNA and RNA are nucleic acids, essential for carrying genetic information. DNA is like the master blueprint, containing all the instructions for building and maintaining an organism. It's typically found in the nucleus of eukaryotic cells. RNA, on the other hand, is more like a working copy of a gene carrying the instructions from the DNA to the ribosomes, where proteins are made.
So DNA holds the long-term information, and RNA is the messenger that puts those instructions into action.
Exactly. And there are different types of RNA, each with a specific role in the protein-making process. You've got messenger RNA, or mRNA, which carries the genetic code from the DNA to the ribosomes. Then there's ribosomal RNA, or rRNA, which is a structural component of the ribosomes themselves. And finally, there's transfer RNA, or tRNA, which brings the correct amino acids to the ribosomes, like a delivery service.
It's like a well-orchestrated dance, with each molecule playing its part to build and maintain life.
And it's all happening at a scale we can barely fathom. But this intricate dance of molecules is fundamental to all life on Earth.
I can't wait to learn more. But I think we've covered a lot of ground for now. We've explored the tools of microscopy, the basic cell types, and even touched upon the fascinating world of DNA and RNA.
And we're just getting started. In part 2, we'll delve deeper into the techniques used to study bacteria, explore the dynamics of bacterial growth, and uncover the secrets of gene regulation.
Sounds like a plan. We'll be back soon with part 2 of your deep dive into the amazing world of microbes. Welcome back to your deep dive. Last time we journeyed into the world of microbes, exploring the tools used to see them, the different types of cells, and the fascinating roles of DNA and RNA. It's mind-boggling to think about all this activity happening at such a tiny scale.
It really is, and we've only just scratched the surface. Today we're going to dive deeper into the techniques scientists use to study bacteria, explore how they grow, and even unravel the mysteries of how their genes are regulated.
Okay, I'm ready. I'm particularly intrigued by the idea of bacterial culture, like actually growing bacteria in a lab. It sounds like a delicate process.
It does have its challenges. Imagine trying to create a perfect environment for something you can't even see. But scientists have developed some ingenious techniques to cultivate bacteria, allowing us to study them in detail.
So what's involved in creating this ideal bacterial habitat?
It starts with the media, the substance that provides the nutrients bacteria need to thrive. There are different types of media, each tailored to specific bacterial needs. Some bacteria are picky eaters, requiring a rich buffet of nutrients, while others are more adaptable, able to survive on a simpler diet.
It's like setting up a microscopic restaurant with different menus for different bacterial diners. But it's not just about the food, is it? What other factors are important?
Temperature is crucial. Just like us, bacteria have preferred temperature ranges for optimal growth. Some like it hot, thriving in environments like hot springs, while others prefer cooler temperatures. Scientists carefully control the temperature in incubators to mimic the bacteria's natural habitat.
So it's all about recreating their ideal environment.
Yeah.
What about oxygen? Do all bacteria need oxygen to grow?
Not at all. Some bacteria, called aerobes, require oxygen, just like we do. Others, called anaerobes, can't tolerate oxygen and will actually die in its presence. And then there are facultative anaerobes, which can switch between using oxygen and not using it, depending on the availability.
Wow, that's amazing. Bacteria are so adaptable. But I'm still a bit baffled by the idea of actually growing bacteria. How do scientists know they're growing the right kind and not contaminating their cultures with other microbes?
Aseptic technique is paramount in microbiology. It's all about maintaining a sterile environment to prevent unwanted microbes from crashing the party. Scientists use a variety of techniques to keep things clean, including sterilizing their equipment, working in laminar flow hoods, and using careful handling techniques.
So it's like a microscopic operating room where everything has to be meticulously clean.
Mm.
And speaking of cleanliness, I'm curious about the different types of cultures. You mentioned pure cultures and mixed cultures. What's the difference?
A pure culture contains only one type of bacteria, a single species. It's like having a monoculture crop where everything is genetically identical. Mixed cultures, on the other hand, contain multiple species of bacteria, more like a diverse garden.
So if scientists want to study the characteristics of a specific bacterium, they need a pure culture.
Exactly. And to obtain a pure culture, they use a variety of isolation techniques. One common technique is the streak plate method, which involves spreading a sample of bacteria across a nutrient agar plate in a specific pattern.
I remember reading about that. It's like a microscopic game of connect the dots, right?
That's a great way to think about it. The goal is to isolate individual bacterial cells so that they can form separate colonies, each arising from a single ancestor. These isolated colonies can then be used to start pure cultures.
Okay, that makes sense. So once you have your pure culture, you can start studying the growth patterns of that specific bacterium. And I see you've included information about the bacterial growth curve. It sounds like there's a whole drama unfolding in that tiny petri dish.
There is. The bacterial growth curve charts the population of bacteria over time, and it reveals some fascinating insights into their life cycle. There are 4 distinct phases: lag, exponential, stationary, and death.
Okay, let's break it down. What's happening during the lag phase?
The lag phase is a period of adjustment. The bacteria are getting used to their new environment, assessing the available nutrients, and preparing for growth. They're not dividing much at this stage, but they're busy synthesizing the enzymes and proteins they'll need for rapid growth.
So it's like the calm before the storm.
Exactly. And then comes the exponential phase, where the bacteria are replicating at an incredible rate, doubling in number every generation. It's a bacterial population explosion.
Wow, that sounds intense. But I'm guessing this rapid growth can't continue forever.
Right. As the population explodes, resources start to dwindle, and waste products accumulate. This leads to the stationary phase, where the growth rate slows down, and the number of new cells being produced is roughly balanced by the number of cells dying. It's like the population has reached the carrying capacity of its environment.
So it's a delicate balance between growth and death. What about the final phase, the death phase?
The death phase is inevitable as resources continue to deplete and waste products build up. The number of viable cells declines exponentially, and eventually the population collapses.
It's a reminder that even in a controlled lab environment, bacterial populations are subject to the same ecological pressures as any other organism. But I'm curious, what happens to the bacteria that survive the death phase? Do they just disappear?
Not necessarily. Some bacteria can form spores. These incredibly resilient structures that can withstand harsh conditions like extreme temperatures, desiccation, and even radiation. They're like tiny time capsules, lying dormant until conditions improve.
So it's like a bacterial survival strategy, waiting for the right moment to reemerge. That's fascinating. But I'm also intrigued by the idea of gene regulation. How do bacteria control which genes are expressed at any given time? It seems like a complex process.
Gene regulation is essential for all living organisms, but it's particularly crucial for bacteria, which need to respond quickly to changing environments. They can't afford to waste energy producing proteins they don't need. One classic example of gene regulation in bacteria is the lac operon.
The lac operon, I remember reading about that. It has something to do with lactose, right?
Yes, the lac operon controls the expression of genes involved in the metabolism of lactose, a sugar found in milk. When lactose is absent, there's no need to produce these enzymes. So a repressor protein binds to the operator region of the DNA, preventing the transcription of the lac operon genes.
So it's like a switch, turning the genes on or off depending on the availability of lactose.
Exactly. But when lactose is present, it binds to the repressor protein, changing its shape and preventing it from binding to the operator. This allows the genes to be transcribed, and the enzymes needed to break down lactose are produced.
So the presence of lactose actually triggers the production of the enzymes needed to utilize it as an energy source. It's like a bacterial feedback loop.
Precisely, and it's just one example of how bacteria can sense their environment and adjust their gene expression to optimize their survival.
That's amazing. Okay, so we've explored the techniques of bacterial culture, the dynamics of bacterial growth, and even touched upon the complexities of gene regulation. But our microbial journey isn't over yet, is it?
Not at all. In Part 3, we'll expand our horizons beyond bacteria, venturing into the fascinating realms of fungi and protists. Get ready for some truly mind-boggling adaptations and ecological roles.
I can't wait. I'm eager to learn more about these often overlooked members of the microbial world. We'll be back soon with the final part of your deep dive. Welcome back to your deep dive. We've already covered so much, from peering through microscopes to unraveling the secrets of bacterial growth and gene regulation. It's like this whole hidden universe has opened up right before our eyes.
It really is a hidden universe. And now we're going to expand our exploration beyond bacteria and venture into the fascinating realms of fungi and protists. Get ready to encounter some remarkable organisms with unique adaptations and ecological roles that are just crucial to life on Earth.
Fungi have always been a bit of a mystery to me. I mean, we see mushrooms popping up after the rain, but what's really going on beneath the surface? It all seems a bit magical.
Well, get ready to be amazed because there's an intricate and interconnected world thriving right beneath our feet. Fungi are eukaryotes, just like us, but they have distinct structures and lifestyles that really set them apart. They exist in a variety of forms, ranging from single-celled yeasts to the multicellular molds that grace our bread and cheese, to the majestic mushrooms that capture our imagination.
So those mushrooms we see, those are just a small part of the story then.
Exactly. They're just the fruiting bodies of certain types of fungi. The real action is happening underground, where an intricate network of thread-like structures called hyphae form a vast web known as the mycelium. And this mycelium can spread over vast areas, connecting the fungal world in ways we're only beginning to understand.
So it's like this hidden network, a subterranean internet for fungi. But how do they get their nutrients? They can't photosynthesize like plants, can they?
You're right, fungi can't make their own food. Instead, they play this vital role as decomposers, breaking down dead organic matter like fallen leaves, wood, and even animal carcasses. They secrete powerful enzymes that digest these complex materials, releasing nutrients back into the ecosystem.
So they're like the ultimate recyclers, nature's cleanup crew. But doesn't that mean that they could be a bit, well, gross?
It might seem that way, but their role in decomposition is essential for maintaining that delicate balance of our ecosystems. Without fungi, the world would be piled high with dead plants and animals. They're the unsung heroes of nutrient cycling.
Okay, I can appreciate that. But I also know that some fungi can cause diseases. I'm thinking of athlete's foot and those pesky yeast infections. So how do those fit into the picture?
You've hit on an important point. While many fungi are beneficial, some can be pathogenic, meaning they can cause diseases in plants, animals, and even in us, humans. The fungi that cause athlete's foot, for example, thrive in warm, moist environments, which is why it's so common in places like locker rooms and showers.
Ah, that explains a lot. And what about those yeast infections? I know they can be a real nuisance.
Candida albicans is a type of yeast that naturally lives in our bodies. It's usually kept in check by our immune system and other microbes, but sometimes it can overgrow and cause infections like thrush in the mouth or vaginal yeast infections.
This is a matter of balance, just like with so many things in nature. Okay, let's shift gears now and talk about protists. I remember from biology class that they're kind of a mixed bag, right?
You could say that. Protists are this incredibly diverse group of eukaryotic organisms that don't fit neatly into the plant, animal, or fungi kingdoms. They include a wide array of organisms, from the plant-like algae that form the base of aquatic food webs, to the animal-like protozoa that can swim around and even hunt other microbes.
Like they're a bit of everything. Can you give us some specific examples of protists?
Certainly. One fascinating example is Plasmodium falciparum, the protist that's responsible for malaria. It has this complex life cycle that involves both mosquitoes and humans, and it's a major health concern in many parts of the world.
Malaria is a serious disease, so understanding how this protist works is crucial for developing effective treatments and prevention strategies. Are there any other protists that pose a threat to human health?
Unfortunately, yes. Giardia duodenalis is another parasitic protist that causes giardiasis, a diarrheal disease, often spread through contaminated water. It's a major public health concern, especially in areas with poor sanitation.
So protisters can be both beneficial and harmful, just like bacteria and fungi. It's amazing how much diversity exists in the microbial world.
It's a testament to the power of evolution and just the incredible adaptability of life. These microorganisms have evolved to thrive in virtually every environment on Earth, playing essential roles in ecosystems and shaping the world we live in.
Well, I have to say, this deep dive into the microbial world has been truly eye-opening. I'm leaving with a whole new appreciation for these tiny but mighty organisms.
I'm glad to hear that. It's been a pleasure exploring the wonders of the microbial world with you. And remember, even though we can't see them with the naked eye, these microscopic creatures are all around us, influencing our lives and the planet in countless ways.
Absolutely. So next time I see a mushroom popping up in my yard or I hear about a new disease outbreak, I'll think of all the intricate processes that are happening at the microbial level. Thanks for taking us on this incredible journey.